4, and so on. So chimps actually have 48 chromosomes and not 46. So I combined chimp chromosomes 2A and 2B into one chromosome. Compare that to human chromosome 2, because that's how they were assembled. So anyways, the peak experiment was basically the similarity for that, the best similarity I could achieve for that chromosome. So on average, it was about 70% similar on average. And actually, my data here on the Y chromosome matched up perfectly with a paper that was published by evolutionists in 2010 on the Y chromosome. So this was the chromosome by chromosome best similarity from the experiments that I did, and it actually varied. That's why I talked about combining chromosome 2A and 2B. So the least similar chromosome was chromosome 4. It was 61.1% similar on average. The most similar was chromosome 22, which was about 77.9% similar. So of all the papers that these evolutionists have published where they focused on a single chromosome to compare to human, which one do you think they chose? 22. Chromosome 22. But when you include all of chromosome 22, it's only 78% similar. So that's a paper I published in the Answers Research Journal on that. And what's interesting is that my data actually, it wasn't anything new. It just really confirmed what other people had already discovered but didn't make the 6 o'clock news. So anyways, this was a paper. It was published by evolutionists out of the Max Planck Institute in Germany, where they actually lined up over 3 million bases of chimpanzee sequence, random sequence. And this is a quote from their paper: only about 2/3 could be unambiguously aligned to DNA sequences in humans. Well, that's close to 70%. And actually, there was a guy named Richard Buggs. He's an evolutionary geneticist, an intelligent design guy, and he actually went back and looked through the chimpanzee 2005 rough draft paper, compared it to the human genome, not with the extensive analyses that I did, but he came up with a 70% and published a short paper on it. And then there was another group out of Italy that developed their own software to compare 10,000 bases from each chimp chromosome selected, 30 base chunks selected at random to human, and they came up with about 70%. So this isn't some wild number that I just came up with. This is confirmed by other studies and other people working in the field, including evolutionists. So what constitutes the 30% difference between chimps and humans? Repeat differences, including higher order repeats, repetitive regions, transposable elements, and there's different categories of these, which are all now proving to be completely functional and critical to the genome. The subtelomere regions, or regions right behind the ends of chromosomes, are unique among humans. They're actually unique among chimps. They're unique among gorillas and orangutans. They tend to be one of the really distinct features of the genome that make each different human or ape taxonomically distinct. And then the protein-coding genes, pseudogenes, which are now proving to be functional, non-coding RNA genes. So all of these things are important things that make up that 30% difference. And evolutionists have known this, but it's just not widely discussed. Here's a paper that was published in 2003 talking about insertions and deletions that occur frequently, what I was talking about, between humans and non-human primates. So they looked at chromosome 21. They looked at about 56% of it, not the whole thing. They found 57 major insertions and deletions that ranged. They ignored all the ones that were less than 200 bases, but they found over 57 that were 200 to 8,000 bases in size, these insertions and deletions, in just looking at half of that. And of course, that was one of the most similar chromosomes that I just showed you in the previous slides. And they were randomly distributed, this was interesting to me, across both regions that contained protein-coding genes and the non-genic ones. So there wasn't a preference for gene-rich regions. They were frequently found in genic intervals, or inside genes, 35%. And they totally avoided looking at 21 megabases of human chromosome 21 that were not highly similar. So studies, and there's been other studies that have been published by evolutionists talking about these huge insertions and deletions. Here's one in 2005. They found 300 base deletions common across the genome, 3,000 plus sites. Here's another evolutionary paper. Insertions and deletions, 15,000 to 250,000 bases on chromosome 16. Here's another one in 2011, 26,509 insertions and deletions, 80 to 12,000 bases in or near genes across the genome. They only examine the gene-rich areas. So this isn't news to evolutionists that are working in the field. They know what's going on. But the general public is constantly told 98%, 98%. So I may kind of just zip through these. But it's turning out that every bit of the human genome and genomes are functional. Retro elements, these transposable elements, or what you may have heard called jumping genes, are now critical to genome function. This was Richard von Sternberg worked at the, where did he work in Washington, D.C.? The Smithsonian. Got fired basically for holding to an intelligent design view of the genome. But he published this paper while he was working there with James Shapiro on how repeated elements format genome function. And it was an incredible paper listing all the different functions and functional features found in transposon. Here's one, 2009, transposable elements in gene regulation in the evolution of vertebrate genomes. Transposable elements rewire and fine-tune the transcriptome, 2013. There's just literally papers published every week now about how important transposable elements are in the function of the genome. Well, it turns out that these are also a major part constituting the difference between human and chimpanzee genomes. So this is just looking at retro elements between chimps and humans. So you can see, and this is just a broad categorization, but you can see the contents are totally different between the two. It contains a lot of very unique retro elements that aren't even found in chimpanzee. So you can see a 71% versus 56% for just the ALU element content difference between humans and chimps. 15 and 25% for the L1s, which is a long interspersed repeat. And then there's other categories as well, totally different in their contents between humans and chimps, yet fully functional in each genome. So ALU elements are particularly interesting to me because they regulate gene expression. They're found in many protein coding transcripts, many long non-coding RNA transcripts. And there's several papers, a number of papers published on that. They're very important in tagging on these long tails on the ends of genes that aid in their regulation in the genome, interacting with microRNAs. They also form circular RNAs in the genome that do regulatory things. So anyways, so this is the human genome lit up for ALU elements in green. So you can see they populate all over the human genome. So I've done some studies comparing ALU elements between chimpanzees and humans. So red is chimp and human is blue. And you can see this from chromosomes 12 to 21, for this particular ALU element, ALU YA5, it isn't even present in humans, but it's all over these chimpanzee chromosomes 12 to 22, and then major differences in their content between all the other chromosomes. And evolutionists can't explain this. It's a complete mystery to them. This is another ALU element, ALU YB9, the same thing, major differences for all the chromosomes. And I've actually done this for orangutan as well, and it actually varies between orangutan and chimp. So this was a paper published by evolutionists comparing the human and the chimpanzee Y chromosomes. 53% of the human genes that are on the Y chromosome, the human Y chromosome, were totally missing on the chimpanzee Y chromosome. Gene classes, entire gene families, 33% not even found in chimpanzee. All you men ought to be really getting excited about this. You know, you've got a girlfriend or a wife that accuses you of being a monkey or something. So I mean, you look at my Y chromosome; it's totally unique. So less than 70% similarity. I love this quote. This is from this paper. Indeed, and this paper was published in a major journal. I can't remember what it was, Science or Nature. Indeed, at 6 million years of separation, the difference in MSY gene content, this is the region they sequenced of the Y chromosome that was gene-rich. The MSY gene content in chimpanzee and human is more comparable to the difference in autosomal gene content in chicken and human at 310 million years of separation. This was a paper published when they sequenced the gorilla genome, and they found that 30% of the genome in gorilla is closer to human or chimpanzee than the latter are to each other. Once again, this mosaic. And I have yet to see a paper on the gorilla Y genome because probably it was the chimp genome was, the Y chromosome was too much of a disaster. So, I know eventually they probably will do it. They'll be forced to, but at this point, in fact, that Nature paper I showed you where they resequenced all those genomes from chimps and from orangutans and from gorillas out in the wild to determine the genetic diversity that was published in 2013. What do you think they sequenced, males or females? All females. Now, why would you do that? If you want the entire genome, you want the Y chromosome in there, right? Of course, that would be bad. Then you have people like me going in the databases and digging that out. So I guess that wouldn't be good, right? So anyways, well, it turns out that the protein coding sequences are also different between humans and chimpanzees. This was a paper published showing that 1,418 of the approximately 22,000 genes at that time were completely missing in chimpanzee. And here's a quote from the paper, which is really nice. I probably just should have read that instead. Our results imply that humans and chimpanzees differ by at least 6% in their complement of genes, which stands in stark contrast to the off-sided 1.5% difference between orthologous, that just means similar in both genomes, nucleotide sequences. So this stuff is in the evolutionary literature. You just have to dig it out. This is another paper published along the same lines where they found 1,285 protein coding genes specific to human, not in chimp. So humans and chimpanzees have not only differences in the gene content, but remember genes have exons, the protein coding regions within them that are split up by introns. Well, it turns out that even the exon differences are pretty significant in between humans and chimps. In my own research, I've found that 6% of time?
Yeah, we're a couple minutes over.
Are we?
Yeah.
Okay.
Can you wrap it up in another?
I will. Yeah, I'll miss all the chromosome fusion stuff, but that's all right. Should we just take questions then? Or?
Can anybody ask a question in 2 minutes or less?
Your slide where you showed the summary of all the chromosomes you compared, was that just on those 40,000 genes that were already turned?
No, that was a separate study after that, where I compared the whole chromosomes. No, the 40,000 genomic segments, that was a preliminary study to optimize my algorithm parameters and to really, that was a preliminary study to dive into the genome comparison thing. So that study basically encouraged me to go on and do the whole genome.